introduce our first speaker, Kyle Vincent, who's going to be talking about weaving Indigenous knowledge and Western science to investigate the impacts of railways on wildlife.
So hi, my name is Kyle Vincent. I'm a master's student at Laurentian University, co-supervised by Dr. Jackie Litzgus and Dr. Jesse Popp. And today I'll be presenting some of the preliminary findings and a general overview of my project on weaving Indigenous knowledge and Western science to investigate the impacts of railways on wildlife. So in general, when we think of transportation infrastructure and interactions with wildlife, most people think of roads. But railways are also ubiquitous across the landscape, and in general, railway ecology in comparison to road ecology has many knowledge gaps. It's understudied when compared to roads, and railways present some unique features that might also present some unique effects on wildlife. So for example, train traffic in general is less frequent than on roads. Trains can't stop as quickly if they do happen to see wildlife. Train tracks are generally less accessible than roads, so fewer observations and fewer visibility from the public. And then the most obvious one is that we have the presence of the steel rails themselves. So you can see in the photo I've included there of a juvenile snapping turtle with the railway in the background. You can imagine that for some smaller species that would present a pretty formidable barrier to dispersal between habitats, for example, for some species. So even though research is limited, we know that railways negatively impact species through things like collisions, barrier effects, and habitat fragmentation. We also know that train collisions have been documented for mammals, birds, reptiles, and amphibians. However, the scope of these studies has been fairly limited, and those really only come from a handful of a small subset, whereas most studies have focused on large mammals. So this project in particular actually was initiated based on concerns from First Nations communities, Shawanaga First Nation and Magnetawan First Nation. And community members there had observed wildlife train collisions and dead wildlife on their traditional territories while out using the land. And they were concerned about the impacts these may have on their local wildlife populations. Both of these communities already have well-established conservation programs, primarily focused at conserving species at risk reptiles. So when concerns came up about those animals in particular, that was what prompted the study. And so I'll go into a little more detail about how we got these quotes, but I just wanted to read a few from community members to put some of their concerns into context for you. So Jerry Smith from Magnetawan said, "You see a lot of turtles, you see snakes over there, you see the odd bear, you see deer sometimes, you see lots of dogs down there that get hit, raccoons. There's a lot of animals to get hit on the tracks. I see all kinds of dead turtles on the tracks, in between rails. They used to get stuck in there. They can't get up and they get trapped between two rails and run over by trains." And so these concerns prompted us to initiate this investigation. And that led us to three main objectives. The first being to use Indigenous knowledge to identify community concerns of railway impacts on wildlife. The second, to quantify wildlife railway use and the resulting mortalities through structured railway surveys and the deployment of motion-triggered cameras. And thirdly, to take that a step further and use that information to identify wildlife mortality hotspots or areas where there's clusters of mortalities or even just crossing events to assess their relationship to environmental variables. So looking down the road, ultimately the goal based on the issues that we identify is to implement mitigation. So that'll help us to determine where to best do that, since those measures can be somewhat costly. And so why is this significant? Well, as I mentioned earlier, railways intersect areas with diverse wildlife, in particular in the case of the study sites that I work at, fairly pristine habitat with not a lot of other development around it, and it's home to a lot of species at risk. So as I mentioned, turtles in particular is one example. All 8 species of turtle in Ontario are either federally or provincially listed as species at risk, and their populations are already in decline. So if these animals are facing risks from railways, as well as many other multitudes of factors, it's definitely something we want to understand in more detail. Identifying species-specific risks could be essential to managing vulnerable species in the future. So like I mentioned, most research so far is focused on large mammals, but obviously the measures for large mammals are not necessarily going to be the same for smaller species like reptiles and amphibians. And then lastly, determining the locations of those mortality hotspots and those factors and landscape features that contribute to them will be essential to developing those effective mitigations in the future, allowing us to target them and hopefully make them as effective as possible when it comes to implementation. And so the first objective was, again, gathering that Indigenous knowledge to, again, paint a more holistic understanding of this issue and also to inform our study design. So in 2019, we did a knowledge gathering session with members from both communities. In total, we had 15 community members from Magnetawan and 17 community members from Shawanaga who volunteered to participate in the study. We conducted semi-structured individual interviews with them with a list of kind of preset questions, but we also allowed the interview to flow naturally in the direction that it wanted to. Those interviews were recorded and transcribed with a transcription software so that they could be analyzed. And so some preliminary findings from those interviews, sort of the highlights were that every participant who we interviewed indicated that they felt that railways negatively impact wildlife and that they were interested in gaining a better understanding of these issues and how they're actually impacting wildlife on the ground. There were observations of a wide variety of wildlife, both using and dying on the railway, and this included things all the way from large mammals down to small mammals, reptiles, and amphibians. It really kind of ran the gamut. But in particular, there was a focus on observations of large mammals and on turtles. And again, I'm going to focus on turtles for today, both based on the relevance for CHS, but also because of their vulnerability, and as I mentioned, species that are already in decline. And so I've included a photo there, a very sad picture of a moose that we found in one of our surveys, and then a painted turtle there as well. So this prompted us to, again, follow up with objective number 2, which is actually quantifying mortality in a structured way with surveys, trying to answer the question, what wildlife interact with the railway and die as a result? So to do this, we had 2 observers conduct visual surveys once per week at each of the field sites in each community to document any wildlife on the railway. When we were walking during this exploratory study, any individual that we encountered, we identified the species when possible, referenced the location with a handheld GPS unit, noted the relative location on the railway, so whether it was in the center of the railway bed or off on either side of the right of way, what condition they were in. So unfortunately, in the images on the right, you can see there's pretty clear evidence of trauma and direct collision with the snapping turtle and the green frog there. And then if an animal was alive, we would also note behavior. So whether it was attempting to cross the tracks, whether it was stuck in between the tracks, or whether it was up basking. And then those observations were grouped by taxon to allow us to conduct the analysis. This is a general overview of the study sites. So you can see both communities are located on the eastern coast of Georgian Bay. We had 1 section of railway at each community, both of them measuring 3.5 kilometers in length, along the same section of single track railway. And this railway crosses a mix of kind of open wetland complexes, mixed forest, and rock barrens. And this provides kind of an overview of, again, what the railway looks like in real life. So on the left here, that's pretty typical of kind of a mixed forest habitat. You can see the single line railway there with the right of way. So when we are walking, we walk on either side of the rails there and search the entire right of way, center rail on both sides. And the photo on the right, you can see the railway sort of in the background there. This is pretty typical of the railway bisecting kind of the open wetland complexes, which are such fantastic habitat for reptiles and amphibians. And so this has allowed me to pooled across all 3 field seasons that we've been able to get out and do these surveys in some capacity, somewhat affected by the pandemic last year. But we've gotten some pretty good results so far. And what we found is that we've encountered at least 39 different species out on the tracks. That's a combination of both live and dead across all 3 years, which strongly corroborates the results that were shared with us in those interviews. Obviously, there's a wide variety of wildlife out there, both using and dying on the railway. On that note, unfortunately, 73% of the wildlife that we were encountered were found dead. So the vast majority were dead rather than alive. And interestingly, 87% of those mortalities were herpetofauna. And so you can see on the right, the vast majority were made up of frogs and toads, which is not totally unexpected based on the same sorts of things we see in road ecology studies, based on relative populations likely, and also their life history strategies and mass movements around breeding time. But what I think is particularly interesting and concerning, and also really mirrors what was shared with us by community members, was the relatively high number of turtles that we found dead along the railway tracks there. making up the second highest proportion of taxa that we found. Even more interesting, I think, are some of the potential causes of mortality that we observed on the railway. So unlike with roads, where typically the majority of turtles that die on roads are actually hit by cars, we found that was not the case with railways. Actually, only 11% of the turtles that we had a confirmed source of death were actually hit by trains, and those were just adult snapping turtles. The majority of the turtles that we found, we believe to have died from heat stress or desiccation. And you can see in the photos there's a little juvenile snapping turtle and a female painted turtle that we suspect to have died exactly like that. While trying to disperse between habitats, they've somehow become entrapped between the two sets of rails and likely died just from heat exposure and time in the tracks. While conducting field work, I do regularly measure the temperature of the railway ties, and it's not uncommon during the peak of summer for those railway ties to be in high 40 degrees Celsius all the way up to kind of the mid 50 degrees Celsius. So you can imagine it wouldn't take very long for a turtle to reach that thermal critical max and lose locomotory function. This is another example of an adult Blandings turtle that we suspect to have met the same fate, and again, showing the signs of potential heat exposure or desiccation, with the head and all of the limbs extended and no other signs of physical trauma to the body. Another really interesting note that highlights the value of incorporating Indigenous knowledge in more traditional Western science approaches was that while we were finding these turtles stuck in between the tracks, and that has been reported in the literature before, we actually had a community member who mentioned a potential cause of death that as far as we know, hasn't been documented in the literature. And that is a turtle actually getting stuck on the tar being leached from a tie. So the railway ties, the wood that they use is treated with creosote and tar to prevent them from breaking down and decaying. And in the summer, these tars kind of leach back out. And so they had observed turtles stuck on the tar and dying. And when we actually went out and conducted surveys, we actually found an example of this. So this was an adult painted turtle that's now been flipped over, but it was basically glued onto the railway tie up in the upper right corner, you can see there. And we've seen other evidence of tar building up on the plastrons of live turtles and some of the other dead ones as well. So this is a particularly interesting result, again, highlighting the importance of incorporating Indigenous knowledge, but also highlighting kind of what a complex issue railway ecology is. It's not just as simple as animals getting hit by trains. There's multiple different factors that need to be thought of and included when looking at things like mitigation. And so our next steps, taking all this information and making it, again, even more useful, particularly for the communities. And so that leads to the third objective, which was hotspot identification. So are the mortalities that we're observing clustered more than we would expect by random chance? To do this, I'm planning on using CIREMA, which is a hotspot identification software commonly used in road ecology studies. And it uses Ripley K statistics to determine those hotspots. So it divides the railway into equal length segments and then simulates random crossing points. It then calculates a hotspot value per segment based on the number of actual observed, again, mortalities or crossings that we see, and then produces an upper confidence limit that highlights those significant hotspots. So it produces that graphically, which you can see on the bottom here. Anywhere where the blue line crosses that upper confidence limit, that signifies a hotspot, and then it presents it almost in more like a map sense on the linear feature up above. Taking that another step further, we're interested in understanding what environmental predictors influence those hotspots. So can we extrapolate from the actual field sites that we're working at? So if we were to try and implement mitigation elsewhere, we would know what landscape features and habitat features influence these areas where we're seeing mortality. And so to do that, I plan on using generalized linear models for frequently killed taxa, and including some spatial variables such as distance to and proportion of land cover in buffers adjacent to those railway segments, using ELC that's been provided by both communities, as well as distance to rock cuts, gap between railway ties, and the leaching tar like we talked about, as I hypothesize all of these may have influence on where we're seeing dead wildlife. And then also some temporal variables that definitely would have an influence on, or potentially have an influence on wildlife movement as well, such as weekly air temperature, cumulative weekly rainfall, time of year, and weekly train frequency. So just want to wrap up by thanking both communities as well as Canadian National Railway and Canadian Pacific Railway for their partnership in this project, as well as the funding that's made this possible. We still have a little bit of a field season left to go, so I'm looking forward to seeing what else we find and getting into some of the other analysis that I talked about. I hope to be able to follow up sometime soon with some more results. Thank you very much.
I want to introduce our next speaker, Claudia Lacroix, who's going to be talking about protected areas as possible effective measures for the conservation of Ontario's herpetofauna.
Hi everyone, I'm Claudia and I'm a master's student at the University of Toronto in the Rollinson lab. And today I'm going to be talking about my fourth year undergraduate thesis and whether protected areas are effective for the conservation of Ontario's herps. So it's no surprise that biodiversity is being lost at an alarming rate. And when it comes to conservation action, one umbrella approach is to conserve the habitats that species at risk are found in. And so shown on the black areas on the map, Canada has a vast network of protected areas. Despite this, protected areas most often don't overlap with areas of high conservation concern. Ontario in particular has some of the richest biodiversity that co-occurs with many anthropogenic pressures. And so I was interested in evaluating whether the effectiveness of protected areas in conserving reptile and amphibian communities by one, comparing herpetofaunal diversity inside and outside protected areas, and two, testing whether this relationship differs between common and species at risk. I use the Ontario Reptile and Amphibian Atlas, which is a citizen science-based initiative to map observations within spatially delimited grid cells, which is shown on the map here. I then overlaid the map of regional, provincial, and national protected areas. We limited the analysis to Southern Ontario, as this area has the highest amount of observations per cell, as well as the most anthropogenic impacts. After removing cells that contained less than 50 observations, the dataset included 1,726 10 by 10 kilometer spatial grid cells, comprising of 397,478 observations and 50 reptile and amphibian species, 26 of which are considered at risk. We ran linear mixed effects models modeling species richness inside and outside protected areas at 3 species scales: all species, species at risk, and common species, and 2 spatial scales. At the core spatial scale, the cell level, we compared raster cells that have a protected area present, shown in green, or absent, which is shown in beige. Then at finer scales, or the boundary level, we removed cells that did not have a protected area present and compared richness inside and outside the protected area boundaries. In our models, we then accounted for such as roads, temperature, wetland, and agriculture cover, protected area size, the number of protected area fragments, and then included eco-province or the cell as a random effect, and then all models were weighted by the number of observations per cell, just to account for sample biases. I found that when it comes to broad scale spatial patterns, there are significantly more species in protected areas. This was largely driven by a significantly higher number of species at risk within protected areas, where common species, on the other hand, did not differ between protected and unprotected regions. When it comes to finer scale spatial patterns, we found that there's significantly less species inside of protected areas. This was the same among species at risk and common species. Now, these results are quite confusing and contradictory to each other. And so does this mean that protected areas are ineffective? Or does it mean that species are perhaps found at the boundary of protected areas? Well, we have to be cautious of citizen science, as the results can depend on sampling effort. And so to verify the results were not an artifact of sampling bias, we took the analysis one step further and ran the same models, except instead of investigating the number of species, if the number of species differed between protected areas, we investigated whether the number of observations differed inside and outside protected areas. When it comes to the cell level, there was no difference in the number of observations recorded in protected and unprotected areas. This means that the observed pattern, where more species are found within cells that contain a protected area, is likely not an artifact of citizen science. Now, when it comes to the boundary level, we found that there's significantly less observations found within protected areas. This means that the observed pattern where less species are found within protected areas is most likely due to a sampling bias that arises from citizen science. And so, are protected areas within Southern Ontario effective? Our results demonstrate that protected areas are at least adequate, although their effectiveness varies among species at risk and common species. This project also shows how citizen science is a great tool for evaluating conservation practices, especially at coarse spatial scales. Freshwater species are some of the most imperiled groups, and so further research on the effectiveness of these areas is imperative. And thanks so much for listening, and I'd like to thank Ontario Nature for letting us use the data, as well as the countless volunteers who added observations to the atlas. Thank you.
So we're going to move on to our next talk, where Fred Schueler is going to be talking about Francis Cook's herpetological papers, and this is a 15-minute talk. So Fred, you can share your screen and take it away.
Okay, here we go. This is a continuation of what we talked about at last year's meeting and haven't done anything organizational about since then. I mean, Francis Cook was my herpetological mentor because I was working in a lab that was all ornithologists when I was a graduate student. And we're living here in Bishop's Mills because there was a way to get a ride into my postdoc at the museum with Francis. So I'm just going to go through some of this stuff and sort of outline the magnitude of the problem. Francis was obsessive about saving everything, and we have photographs here of dumpsters that Joyce has filled with paper that she considered wasn't significant from stuff that he had saved. He recorded the menu at every meal that he ate and saved all their income tax stuff all the way back. And
every scrap of correspondence.
Every scrap. Let's see if this page down. Down here, it doesn't seem to.
No, how about that one?
Oh,
Next page. This isn't working.
all right. How about that? So there's my
Okay.
abstract, which you can read on the website. So when Sherm Blakeney, who's standing here, retired from herpetology in 1959, Francis, who'd been his assistant, became the curator of herpetology at the National Museum. And he was, when I was a graduate student, I was told that there was one professional herpetologist in Eastern Canada, and that was plenty. And
By Francis.
No, this was by Ed Crossman at the
Ah.
ROM, where they were trying to keep herpetology out of the ichthyology and herpetology section so they could do more stuff with fish. So Francis was, he was the center of Canadian herpetology for many years, and then in 1993, with some weirdness at the then Canadian Museum of Nature, they fired all the curators of groups that the public would consider animals, and that included reptiles and amphibians. And Francis went on to be a research associate at the museum, and he brought home a lot of the files of the herpetology section that he was afraid would be thrown out because there'd been a bout of document discarding at the museum. And so here's pictures of, and so Joyce and her children, Francis's children, have been going through first his study, which is what we talked about last year, and then the drive shed, and they still have the barn where he's got bins and bins of manuscripts from when he was editor of the Canadian Field Naturalist. And that's just sort of an introductory picture to the volume of documents that we're talking about. And here's our, I mean, we're not exactly alien to the idea of saving a whole lot of stuff. This is our museum trailer with the remains of the Eastern Ontario Biodiversity Museum, but all those tubs are documents from from the drop shed, 68 tubs, Rubbermaid tubs.
In
of-
addition to the 25 that are at the North Grenville Historical Society right now in Kentville.
So as I said, these include the files of the herpetology section that Francis brought home and the museum closed down herpetology in 1993. And so some of the things that are included here are, Francis started to work on a Reptiles and Amphibians of Canada book in 1960, and he was very secretive about a lot of stuff, and nobody has any real idea how far he got towards having a manuscript. But these are some of the folders the binders he had for information for that book. And so we've, in addition with the documents, we've sort of inherited the idea of going through and finishing Francis's book.
And I've done the watercolors illustrations of the live specimens of nearly all of the amphibians and reptiles of Canada.
And this was in the 1970s before we knew about non-acid papers. So these papers,
Some of them, the
some of
older
them.
ones. Yeah.
And then there's measurements, and so there's live, fresh measurements of basically all the specimens that Francis collected that are in the museum. And when it turns out that, I don't know how familiar people are with the book called The Owl Pen, but this was the story of a couple that bought a sort of rough farm in Simcoe County back in the 30s, and the story of how they moved in there and made it into a place where they raised a lot of honey and goats. And this turns out was the inspiration for Francis when Francis came back from being from doing the coursework for his PhD at the University of Manitoba. And so as soon as they moved in there, Francis started to do a lot of monitoring of the seasonal movements and the size and growth of reptiles and amphibians, both around their home and at a series of different ponds in the vicinity. And also he had a system of auditory stations where he recorded calling by anurans. So that's I mean, we haven't gone through all these 75 or however many bins of stuff. We've just sort of gotten them to where they're safe. And there's a lot of historic correspondence. We have stuff that goes, we have a pile of photographs that Clyde Patch did in the 1910s. And there's this, at least Shelley Lozier's files, herpetology files from the Royal Ontario Museum, which Francis salvaged to the National Museum when the ROM was in their non-herpetological phase. And those are the ones, I used those when I was writing the hognose status report, because Lozier had a whole lot of correspondence about hognose snakes in various parts of the province. And I'm told that there's a whole half bin of Francis's letters to me. And so there would be corresponding stuff with many of the rest of us. And then there's the National Museum data sheets, some of which are still held at the museum, and some of which
I'm sure he had photocopies of all of them.
Some of them, the ones from around his home, I don't imagine are in the museum. These are obviously photocopies because the sheets were yellow. And then there's whole bins full of historic herpetology publications, and bins full of books, and just goodness knows how much else. Now,
There was also the time that he was editor of the Field Naturalist, and all the correspondence he had with the authors.
Right.
He saved everything.
And so anyway, Aleta and I have a certain amount of experience with this because when Wayne Grimm, the land snail guy, died, we managed, he'd already made contact with Canadian Food Inspection Agency to publish his introduced land snails manuscript. And with Robert Forsyth and Isabel Picard, we wrenched it into a book, and we got his papers and specimens into the Canadian Museum of Nature. So if there's some kind of organized movement for Francis's stuff, which is about 8 times the volume of what Wayne's was, we're game to go ahead, but it's more than we can handle by ourselves. And this is highly speculative, but this is a place on our land where we back in the day we were going to build a museum building in this clearing, but it's been 30, 35, 20 years since then, and stuff has grown up, but it could be, it's a site that could be cleared and a straw bale or some other kind of building put up to work on all the stuff that Francis has left behind, and all the data. Francis's publications were mostly range extensions, except for his thesis, were mostly range extensions. And so there's lots and lots of data that could be analyzed to actually test hypotheses or track changes through time. And we'd really like to see some organization larger than just us taking up some of the burden on this.
And one of the things that's required is some space for laying things out, because as you saw in our museum trailer, there's barely even an aisle left for walking from one end of it to another. So yeah, it's very exciting. Whenever we open any of these bins, it's always really fun to see what's there.
Okay, so here's a bin that has some of Francis's field notes and has Sherm Blakeney's field notes from 1952, 53.
1958 to what?
I know some of them are
1954 to 55.
And these are field notes that are so antique that the measurements are written in inches. So so that's it. I will send a notice around by email asking people if they want to be on a list of people who are concerned about this, and maybe develop a plan. But we don't really have a plan right now.
If I can add, one of the things that needs to be done is Francis's work on his book, Amphibians and Reptiles and Amphibians of Canada, with the illustrations that are actually being held by the museum at this time, should be continued and concluded. And so we need some collaborators on that, and some people to take charge of particular taxa and the species descriptions and ranges and stuff on these taxa.
So we're going to move on to our next speaker, who is Aidan Maloney, is going to be talking about understanding the impacts of wildfire and wind farm construction on the herpetological community of Eastern Georgian Bay. Aidan, take it away.
So hi everyone, my name is Aidan, and today I'll be discussing some research being investigated in collaboration with the Litzgus lab at Laurentian University and Blazing Star Environmental. And what we're examining are the impacts of wildfire and wind farm construction on the herpetological community of Eastern Georgian Bay. So before I begin today, I'd like to do a brief land acknowledgement. So for me specifically, I wish to acknowledge how grateful I am to have the opportunity to live, thrive, and to be able to perform research on the lands of Hemby Inlet First Nation. So Eastern Georgian Bay is a recognized hotspot for common and at-risk reptiles and amphibians within Ontario. However, this is an area that is under pressure for development, thus impacting the native herpetofauna. Hemby Inlet Wind is the largest single-phase wind project in Ontario and is the largest First Nations wind project in Canada. Construction began in 2017 and the site became operational in 2019. During the summer of 2018, a large wildfire occurred that affected areas adjacent to and within the footprint of Hemby Inlet Wind. We aim to examine the effects that these combined disturbances have on the herpetological communities within the area. More specifically, we wish to quantify the abundance, diversity, and health of Inuran, snake, and turtle individuals and populations in 4 replicated site treatments: control, wildfire, wind farm, and wildfire plus wind farm. Within each of these 4 treatments, we will have 3 replicates for a total of 12 sites. Since this is an ongoing project that is still in the data collection phase, I'll mainly discuss our methods. So we will conduct coverboard surveys to gain a representation of snake diversity, abundance, and health within our treatment areas. We will deploy clusters of 4 coverboards per site, with 2 being plywood and 2 tin to maximize potential preferences smaller northern snakes may exhibit as per the findings of previous research. Each cluster will be surveyed once a week from May to August. Snout vent length and body mass of each snake will be taken to use for body condition indices. This figure represents how we plan to cluster the coverboards at each site that will be placed approximately within a 3-meter diameter. Inuran course recordings will be conducted with the use of acoustic recording units. This will be done to obtain a representation of Inuran diversity within each of our treatment areas. Each recording will be rotated between each of their associated replicates every 3 days. Marker capture will be done to determine turtle diversity and abundance. Morphological processing will also be done to obtain body condition indices. Now I'll briefly discuss the analysis that we plan on performing. So the main diversity analysis that we will be performing will be done with the use of the Shannon-Winer Diversity Index. And this is a measure of diversity that combines species richness and their relative abundances. Program Mark will be used to analyze our marker capture data. This is used as a way to produce survival and population estimates, which can then be used in a population viability analysis. We will be using an open model for these analysis. Body condition indices will also be used to estimate the condition of individuals based on the relationship between body mass and measures of length. A similar study from the Litzgus lab examined the effect of smelting impact on a herpetofauna community ecology within Sudbury. The x-axis represents the average number of combined individuals per treatment, while the y-axis represents species richness. Like our study design, this research consisted of 4 treatment areas with 3 replicates within each treatment zone, with reference representing the least impacted and barren representing the most. As you can see with these figures, for both reptiles and amphibians, species richness and abundance drops as the smelting impact increases. We're expecting to see similar results as these, and we'll base much of our analysis and experimental design to how this research was conducted. Our plan for this research is to be a long-term 20-year study that I will analyze for the first 5 years. So what we're hoping is that this research will inform implementation of mitigation strategies for future large-scale wind farms, and this is especially in areas prone to wildfires. Not only that, but this will help us understand community and individual level impacts on local herpetofauna. More importantly, however, as we transition to green energies, such as wind energy, we need to understand how wildlife are affected and how we can decrease these impacts. Thank you everyone for listening, and I'll be happy to answer any questions at the end.
So our next speaker is Emily May, the last speaker in the session, and the presentation is on the elemental content of reptiles and amphibians, nutrient cycling consequences.
So I'm Emily May. I'm a PhD candidate in Dr. Rana El-Sabawri's laboratory at the University of Victoria. And I'm going to be presenting about a subset of the results of a meta-analysis I recently finished on the elemental content of vertebrates. And specifically, I'm going to talk about the phosphorus content of reptiles and amphibians and some of the consequences this might have. So I wanted to begin by talking about why vertebrates are important in nutrient cycling. My main interest is how vertebrates recycle nutrients through their diets, because this can be directly related to their elemental content. So when an organism eats, it partitions dietary nutrients into different roles. Some nutrients go towards respiration and general metabolism, but some are sequestered in new tissue. And some of these are, which removes these nutrients temporarily or permanently from the pool of available nutrients. But the animal will always produce waste products, which will recycle some of their dietary nutrients back into the ecosystem. And so in this way, animals are acting simultaneously as a source and a store of nutrients. Compared to most other animals, vertebrates specifically are large and long-lived and mobile, and they're relatively rich in important nutrients like nitrogen and phosphorus. And this makes vertebrates long-term stores or sinks of nutrients in their respective ecosystems. And they're also vital in transferring large quantities of nutrients between ecosystems. So we see this when anadromous fish transfer enormous quantities of marine nutrients from freshwater to aquatic and terrestrial systems. And we also see it at a different scale when amphibians transfer aquatic nutrients to terrestrial ecosystems during metamorphosis. So why is elemental content important in vertebrate ecology? The reason why I'm interested in it is that elemental content is a super interesting metric for understanding how animals contribute to nutrient cycles. And the first reason for this is that elemental content provides information about how organisms have accumulated nutrients over time. So this shows the extent to which this animal has prevented others from taking up these nutrients. And second, it shows how these animals may take up and recycle nutrients in the future. So an animal with a nutritionally expensive trait will likely need to take up more of the nutrient in question and may recycle less in their waste than an animal without the trait. And elemental content, therefore, is summarizing all of these nutritionally expensive traits and can potentially act as a predictor. So when you're interested in vertebrate elemental content, you have to consider bone, as it's what makes vertebrates so high in phosphorus compared to invertebrates. So the mineral structure of bone is primarily composed of calcium and phosphate. So bone is a huge contributor to the phosphorus pool. Also, bone decomposes really slowly, which increases the potential for vertebrates to act as nutrient stores even after death. So for most of my study, I made predictions about the percent phosphorus of vertebrates based on the bone composition in different vertebrates. So my goal was to compile and analyze the currently available measures of phosphorus content in vertebrates with the goal of assessing how life history traits and taxonomy affect whole body phosphorus. And I began with a systematic review using these keywords, and then I used a screening process to determine which sources were suitable. So to be included in our study, papers had to include the whole body percent phosphorus of a non-fish vertebrate, because fishes are very overrepresented in the literature. And so we excluded many papers as well that only included part of the body, or sort of things like stable isotopes and things like that. And the paper also had to be published after 1950 and had to include a measure of variance that we could use for study weighting in our statistical models. So ultimately, we collected data from 39 amphibian species and 17 reptile species. So within amphibians, there were a few taxa that appeared especially favored, especially plethodontid salamanders and ranid frogs. But there was little repetition in reptiles, so most reptile species in our data set are represented only once. So these are the variables we collected from each of the studies we found, where possible. So our response variable was whole body percent phosphorus, and we looked at the impact of class and order, as well as the impact of life stage, habitat, and diet. I really wanted to look at size and sex, but not enough people included sex-separated data. And I haven't presented size here because it had a very small effect when the effect of life stage was already accounted for. So it is slightly confounding and not super useful to look at here. So I'm going to discuss our hypotheses now. So these hypothetical plots I'm showing just show increases in phosphorus content. So our first two hypotheses are about taxonomy. So for class, we hypothesized that amphibians will generally have much lower percent phosphorus as they have modified skeletons that often have low mineral content. So reptiles will largely have higher percent phosphorus than amphibians. And this translated into the following prediction for order, with anurans generally having the lowest percent phosphorus, followed by salamanders, squamates, and then turtles. So anurans and testudines have the most modified skeletons, with anurans having low mineral, high flexibility skeletons, and testudines having the opposite, which is why they've been placed on the opposite sides of the spectrum. Our next characteristic is life stage. So because life stage corresponds with bone development, we predicted that percent phosphorus would increase with age. And this is likely greatly increased in terrestrial frogs who must transition from an aquatic to a terrestrial ecosystem. So in the same vein, we predicted that terrestrial animals will have higher percent phosphorus than aquatic because terrestrial animals must develop a highly mineralized skeleton to combat gravity, which is less necessary in a buoyant medium. And finally, we predicted that organisms with diets higher in animals, which are nutrient rich, would have higher percent phosphorus than organisms with diets higher in plants, which are carbon rich. So a carnivorous species may be more likely to maintain high bone mineral density, which would increase its percent phosphorus, or it may be more likely in general to evolve a higher phosphorus body as it would be less costly. And that was said, we ran this global model, which is percent phosphorus given class, order, life stage, habitat, and diet. And so moving on to my results. Before I go into my actual statistical results, I just wanted to show you how reptiles and amphibians varied overall. So I plotted the percent phosphorus against class here, and broadly speaking, reptiles, which are on the right, had a much higher percent phosphorus than amphibians, which are on the left. So reptiles had a mean percent phosphorus of 3.08%, whereas amphibians had a mean of 1.74%. So it's quite different. And this difference is even starker when we're only looking at adults. So amphibians had a mean percent phosphorus of 2.11%, and reptiles had a mean of 3.76%. However, you will notice that there's quite a bit of spacing between reptile measures, and this is partly because the sample size is quite a bit lower, but there's also another reason which I will go into later. So our best model from all of our from our global model was percent phosphorus given order and life stage. So when we looked at the effective order, you'll see that it largely mirrors this prediction we had, where anurans have the lowest and testudines have the highest percent phosphorus. So here is a plot of our order results for percent phosphorus. So anurans on the far left have the lowest percent phosphorus, while testudines, which are the second to the right, have the highest. Squamates have slightly higher percent phosphorus than the two amphibian orders, but is nowhere near the difference that we see with turtles. So in our models, turtles largely drove this entire order relationship, which explains why most of the effect of class was driven by order rather than by an actual whole class difference. So the difference is even starker when we remove life stage and only look at adults, as our adult testudines had a mean percent phosphorus of 6.16% compared to squamates at 2.9%. So adult anurans and iridelans were actually really similar with means of 2.08 and 2.28%. When we look at the effect of life stage overall, we can see that percent phosphorus increases with development from larvae and newborns to adults. So larvae and newborns are on the right, and then if you go to the left, it's increasing age. However, the magnitude of this relationship was high, really high in anurans, and was not as intense in other taxa, including salamanders, which was super interesting. So for reference, here's the plot with only anurans included. So not only did we see large differences between life stages, but we also had a fairly large sample size to look at this compared to our other relationships, which might explain part of it. So to summarize, elemental content in our data set was largely driven by order and life stage, with small effects of other variables. The effect of order was largely driven by testudines, who had a much larger percent phosphorus than other reptiles and amphibians, while the effect of life stage was largely driven by anurans. So what does this actually mean for nutrient cycling? So very generally, we can see that turtles are concentrating much more phosphorus than other vertebrates. And this is hugely significant because of their long lifespans, as well as their presence in both aquatic and terrestrial ecosystems. So I'm kind of thinking of them as the phosphorus storage old growth trees. The significance of life stage, especially in amphibians, may be essential for cross-ecosystem nutrient transfer, where tadpoles of terrestrial frogs are accumulating aquatic nutrients, but then remove them when they metamorphose. And if we're looking at this more broadly, the removal of reptiles and amphibians may have devastating nutrient cycling consequences. So unlike birds and mammals, reptiles and amphibians are ectothermic and so can create biomass with much greater efficiency. And this combined with their often long lives can make them valuable nutrient sinks in a lot of ecosystems. So removing them or reducing their numbers will fundamentally change how nutrients are cycled. So for example, it may lead to altered limitation regimes due to changes in nutrient availability or community structure. So this was sort of briefly looked at by Wenger et al. In 2019 in their paper called The Missing Dead, where they quantify the effect of vertebrate removal on nutrient input into rivers. So one of their case studies looks at how the reduced turtle populations may reduce phosphorus input to freshwater ecosystems, as large populations of turtles would have once provided a fairly steady stream of phosphorus into these ecosystems as they died and slowly decomposed. So the effect of nutrient accumulation in vertebrates can be both interesting and essential for nutrient storage and recycling. So with that, I'd like to thank you for listening. And I'd also like to thank my graduate supervisor, Rana El-Sabawi, for helping me plan my project. And I would like to thank my sister, Erin May, who is a librarian and helped me with all of the systematic review things. And I also wanted to acknowledge a couple of researchers who gave me extra data sets for this project. And because we didn't find much data, I'd really love to do a project that characterizes percent phosphorus of additional reptiles and amphibians. So if anyone has any ideas on this and how I can do this without having to kill any additional animals, so things like roadkill or laboratory animals, please send me an email. I would really appreciate that. But with that, that is the end of my presentation.
So I'm going to start with a question for Claudia from Julia Riley, who asked, do you think that survey effort is similar between unprotected and protected areas? May that affect the observation rates you analyzed? Awesome work.
Yeah, so yeah, I just answered that in the chat, but no, just to summarize, that's a great point. There's quite a bit of observations, much more observations outside of protected areas, but that's probably largely because there's so much land that's unprotected. But when you look on like a per cell basis, there's definitely more observations within protected areas, although survey effort obviously varies. And so I'd imagine that protected areas have a larger survey effort. But yeah, it's super hard to disentangle actual effort with this type of data.
Great, thanks. So I'm going to keep working through the chat, but if you have a question that you want to ask, you can raise your hand and I will alternate between the chat and folks that want to ask them. We're going to move on. A question from Gabriel for Emily. Considering you're looking at the nutrients being stored in herps at varying life stages and diets, wondering if you could incorporate ecosystem services into your research to reach a wider public audience and conservation impact. Nutrient cycling is exceptionally important. Ecosystem services, especially related to water, and are super relevant in a big field of research right now. I feel like your research could provide some really useful input and information on this field.
Yeah, that would be super interesting. We're working on sort of figuring out more applied uses for a lot of the research that we're doing, because we do a lot of phosphorus analysis on other animals in our lab as well. So I'd be super interested in doing that as well. Yeah.
Okay, I don't see any hands up yet, so I'm going to move on with another question. I have a question for Kyle about what sort of mitigation you would use for rail road mortality or rail mortality railway.
Yeah, it's a very good question. So the actual mitigation technique itself is a little bit outside of the scope of my study right now, but based on the literature that I've read, there are a few places where people have tried mitigation. It seems fairly successful, and ultimately I think it would be fairly easy. Ultimately it comes down to basically like little mini underpasses under the railway. That's been trialed in Massachusetts and Japan, among a few others. And there was a study for gopher tortoises where they did essentially the same thing too. So I think ultimately it would be potentially fairly easy. It's just a matter of looking at some of those methods and then actually doing significant enough follow-up to make sure they're actually being used. But yeah, there definitely are a few techniques out there. Just in comparison to roads, it's understudied for sure.
Great, thanks. We have another question in the chat for Emily from David, asking, are you also able to assess other elements such as nitrogen?
Yes, that was actually part of my meta-analysis. I just wasn't able to present everything here because I didn't have the time. But I did nitrogen, phosphorus, and the nitrogen to phosphorus ratio. And I have some carbon data as well. So yeah.
Kyle, it looks your data. I don't think your data is any different from the last time we talked about this in the lab meeting for snakes. Hey, I have a question about snakes.
Yeah, I'm just trying to remember. It might be a little bit different, but the ratios are all pretty much exactly the same.
Yeah, so I think this is kind of interesting because, I mean, so in the study that I presented, when we looked at habitat suitability, we actually classified rail right-of-ways as relatively high suitability relative to compared to other land cover classes. So based on some literature that we had, we assumed these were actually quite suitable for Massasaugas, particularly in situations where open habitat, like a low canopy cover habitat is limiting. And it looks like your data so far, I mean, there isn't a whole, it doesn't seem like there are many samples of snakes, but it looked like there were about 5 dead snakes out of 40. Looked like 88% of your observations were alive. So I'm wondering if something that might come out of this is information on the permeability of railways or suitability of railways for snakes, which might show that they are not really that much of a threat for snakes and actually provide like usable open habitat. I'm not sure if there's any insights on that yet with what you have.
Yeah, for sure. I think there's definitely potential for that. It's certainly not all doom and gloom for every species when it comes to railways. And like you say, with snakes, again, overall, our sample size is fairly low for snakes across the board, but they're one of the most common live observations that we do have. And at the same time, one of the lowest. So I think you're probably right that that open, like the railway ballast does present fairly suitable habitat for them. There's a few, again, one-offs. We did have one dead Massasauga that we actually just got in a survey recently that I don't think would have even been included in that data that I presented because it was a little bit of an earlier analysis. But I think most snakes have a capacity to evade a lot of the threats that railways present. With the case of that one Massey, it was right in the center tracks, and based on the injuries that it had to it, I think it might have actually been trapped in between the tracks and tried to strike at a train as it was going over. So probably wrong place, wrong time. Most of the other mortalities that we've seen have either been smaller snake species like red bellies and ringneck snakes getting crushed in the ballast just off the side of the track. So I think they're still using it as habitat and just as a consequence of the pressure of the trains going over being crushed. But I think ultimately on the whole, again, small sample size, but seems to be more live observations for sure for snakes than dead. But there's a lot more to unravel there, I think, too, that we might be able to kind of tease apart.
Thanks, Kyle.
No problem.
Okay, great. Thanks. We're going to do one last question. I see, Patrick, you have your hand raised. Go ahead, Patrick, and then we're going to wrap up this Q&A session.
Thanks to everyone in this session for your talks. Really, really interesting stuff. Kyle, I actually have 3 questions for you, so I might have to triage here. So Baxter, Gilbert, Riley, and colleagues a few years ago did a paper about insect road mortality surveys, and they extrapolated based on the number of insect road mortalities they found, what insect mortality might look like across the roads of Canada or Ontario, Canada, Eastern North America. Would you have any plans to extrapolate your data on railways and where you find hotspots to a much broader geographic scale?
Yeah, I think that's definitely, that was an excellent paper. I don't know if James is listening, but it was fantastic. I think there's definitely potential there. It's not something that I have delved into too much yet. So far, I'm mostly focusing on like the specific study sites that we're working on. But I think when I start to get into that more kind of landscape level, habitat feature and landscape level analysis, I'll be able to get a better look at that in general. In terms of like mortality rates and things, we're a little bit limited because we are only able to get out once a week. I know with that study, the sampling rate was a lot higher. So in terms of like getting accurate numbers for things like that, we'll see how things come out in the wash. But I think there's definitely potential for some extrapolation with kind of large scale land cover classes. But I think it depends on the habitat too. The areas that we're working in, especially for reptiles and amphibians, it's pretty ideal for them. So definitely something to look at for sure.